Dustin Tyler from the Department of Biomedical Engineering, and it's officially about 5 o'clock by his clock. So we've got enough to get a little coffee to get him started. So we introduce today Professor Philip Sabes from University of California in San Francisco. His work is really focused on discovering the physiological mechanisms and the computational processes or principles by which the information is processed in the cortical sensory-motor circuits, and particularly looking at how the experience reshapes and essentially learning in these circuits and things. His pedigree includes MIT and the Salk Institute in his training, and he's currently the Jack D. and Dolores Lang Endowed Chair Professor of Cell Physiology at University of California, San Francisco. And he publishes regularly in journals such as Nature Neuroscience, Journal Neuroscience. It's also, the interest for me is basically a good core understanding of neurophysiology and how interaction with the environment and continual feedback, as well as electrical stimulation and electrical interaction with the brain, shapes that experience over time and ends up the connection. So from basic neuroscience, does a lot of work in macaque monkeys to where our interest is how it can maybe translate some of that work in the electrical stimulation things that we do as well. So I'm looking forward to a very interesting talk, and I'll let him go from here. So Professor Sabes, thank you.
Thank you for that introduction, Dustin, and to Bob and Dustin for inviting me, and also to the Department of Neurology for hosting the seminar. So the work I'm going to talk about today was the work of a large number of students and postdocs in my lab, and this is just sort of a full list of people in the lab, but I will highlight those that did the specific work when we get to it. I also want to mention DARPA. DARPA funded most of DARPA funded the last part of this talk, which is the stimulation part, and it was under the repair program, which really gave me a great opportunity to try something that was a little bit outside the box and quite different from what we had been doing before. And I hope you'll agree that it worked out quite well, and so it was a really great opportunity for me. So what I want to do today is I want to tell you a little bit, just briefly at the beginning, about the importance of somatosensation for normal movement control and also for brain-machine interfaces. And then I want to tell you about our approach that really is driven from, at the beginning, the basic neuroscience. So I want to tell you a little bit about the role of sensory estimation or sensory feedback in motor control. And then at the end, I'll tell you about our recent work applying that. So to get started, this is a video that some of you may have seen before. I'm going to just go ahead and play it.
With no sense of touch below the neck, and unable to sense how his limbs are moving. Charles is not paralyzed, but he has lost his proprioception, the hidden sixth sense that tells the brain where the body is in space. Proprioception is so basic to our experience of being in the world that most of us are unaware that we have it.
Okay, so that video, I think what's in some ways most striking about that video is that if I were to have turned off the sound, and if you didn't know what my talk was about, and you had seen just this video, obviously that was in slow motion, but this video of somebody struggling that hard to simply get out of a wheelchair and sit on the ground, you would assume that the person had some sort of motor deficit. But in fact, this person's deficit is entirely a sensory deficit. He's lost a sense of proprioception, knowing where our body is in space. And by the way, this is from a really interesting BBC documentary, which you can find these days on YouTube. And the point of the documentary actually isn't about him. It's about another guy who's managed to train himself out of this, who's managed to learn how to recover motor control despite having no proprioception. It's really quite fascinating. I recommend it. But in any case, our ability to move in an effortless manner really depends on having feedback from our bodies. So Claude Guez and Bob Sainberg years ago did some very nice work on this topic and showed that even very simple movements, like they were doing something like a slicing movement, like imagine taking a knife and kind of going like this in front of you to slice a piece of bread. Normal subjects can do this quite easily and make very straight movements there and back. But if you take a subject who's been deafferented so they can't feel the position or posture of their upper limb, this is what you see. Movements that are curved and have trouble making that quick reversal. So this is, again, a very simple movement that's impaired because of the lack of proprioception. Now, we think that when you look at performance in brain-machine interfaces, it's not quite where we really want it to be. So this is a video from the Pittsburgh group that is, I would say, probably state-of-the-art brain control of an external device. So this is a human with brain gate interface. That's 100 electrodes in her motor cortex. And she's controlling this robotic arm, which is trying to do this task of pouring the ball into another cup. Now, when I see that video, part of me says that's just amazing. That's unbelievable. It's really great performance, dexterous movement, accomplishing a task. That's really fantastic. On the other hand, I think that's not natural movement. That's not where we want to get to. We want to get to movement that's as fluid and effortless as what you can achieve with your own body. Now, the question is, why is that not at the level of natural movement? Now, surely there's engineering work to be done, better neural interfaces, more neurons, better decoding, and so on. I'm not saying that we've maxed out what can be accomplished on the engineering side, but I think that there's another component that's really important, and that is this person, when doing that movement, is essentially deafferented. There's no proprioceptive feedback. And there's a hint that providing proprioceptive feedback in the context of BMI could help recover naturalistic levels of movement. So this is work from Aaron Szyminski and Nico Atsopoulos' lab at Chicago, where he took a monkey who was doing sort of a classic simple BMI control task. So there are 3 versions of the task. In this particular case, the monkey had his arm in a little 2-dimensional sled, a little exoskeleton, and was moving naturally his arm from one point to another. So that's visual control. That's normal movement control. And then in the second case, he was doing what we think of as classic BMI control. So his limb was stationary, but the experimenters were reading out neural activity from this monkey's motor cortex, using that to decode the intended movement and moving the cursor along the screen from spot to spot by decoding that neural activity. So that's BMI, brain-machine interface mode. And that's with visual feedback only. So the monkey can't feel that dot, but he can see where the dot's going. And then the third version was they had the monkey do the same task as in the middle here, where the monkey was getting visual feedback and moving the cursor with neural activity. But they were passively moving that exoskeleton, that sled, around with the monkey's arm, so that the monkey was now getting both visual and proprioceptive feedback. Again, passive movement, but the monkey was getting proprioceptive feedback. Now, if you look at performance, with natural movement, this is sort of the baseline. This is where you want to be in blue. And then, obviously, with BMI mode, performance is not as good, as I showed you in the previous video. Monkeys and humans are not as good with BMI control. But when you give proprioceptive feedback to this animal, performance gets much closer to that you would want to see for natural movement. So again, this is just an indication that if we could provide artificial feedback in the context of brain-machine interfaces, that we might get more naturalistic behavior. And not only that, there's this sense of ownership or embodiment of the device. And that's something that Dustin and I were talking a lot about yesterday and with his lab, that once you start providing a somatosensation to a device, people start to feel like that device is more part of them. And not only will that increase use, but it sort of increases the qualitative satisfaction of having the device. It makes it feel more like you have your arm back. Okay, so the question is, how can we provide proprioceptive feedback in the context of a real brain-machine interface? Now, the analogy I think that I like to think about, that in some ways was really the technology that drove brain-machine interfaces, is the cochlear implant. So this is in some ways kind of the canonical success story of how it is that you can take sensory feedback and provide it artificial feedback back to the nervous system. And the cochlear implant, I think the 2 hallmarks to think about in this context about the cochlear implant and why it was able to work so much earlier than any of the other devices that were developed. The first is that the cochlea has a very nice tonotopic representation. So this topography of mapping of the stimulus space to the anatomy makes it very easy, because if you can put an electrode down into the cochlea, of course, if you stimulate a particular electrode at a particular location in the cochlea, you can have a pretty good idea of the nature of what the subject will hear. And the second thing about it is that because of this topography, it was potentially possible to create what we call biomimetic stimulation, which is to say to recreate patterns of activity that are similar to, or as close as much as possible, replicate the patterns of activity that would be seen in the normal environment. Now, of course, if you only have 8 or 16 or 32 electrodes, you're not going to be able to completely recreate the patterns of activity that you get during normal sound in an intact cochlea, but at least you could be working toward that biomimicry, working towards patterns that recreate natural patterns of activity. So I think these are the hallmarks in my mind of the cochlear implant. So does that approach work for artificial somatosensation, and in particular for artificial proprioception? Well, I would argue that there are some very large hurdles in the way of applying that kind of approach to artificial proprioception, at least when it comes to interfacing with the brain. And the first is that the topography is not clear. So both within the peripheral nerve and within the brain areas responsible for proprioceptive feedback, the nearby neurons or nearby nerve fibers can encode very different things. This is something that Paul Moresco and I were talking about yesterday, that if you go into the areas of, say, the rodent brain that encode muscle spindle activity in a rodent, you can see nearby neurons encoding completely different muscles, even for distal versus proximal musculature. So that means that if you go in and you stimulate, you can't create a simple unified percept of, say, for example, my shoulder moving. You might create a more complex representation. And that's a problem for recreating natural patterns, biomimetic patterns of activity. Secondly, you can't really invert the biophysics anyway, so stimulating extracellularly isn't going to give you naturalistic patterns of neural activation. And finally, in the clinical setting, it's not clear how this works, because if you have an amputee or if you have spinal injury, there's no way to get that mapping. So if you look at people like Sleeman Ben Smai at Chicago, who's just done some absolutely beautiful work looking at somatosensory organization in somatosensory cortex, say tactile responses of the digit, and then using that to stimulate and recreate patterns that make the monkey feel like he's feeling his digit being tapped. That's great. But of course, in order to do that, he first had to map out those representations. Now, that may be more practical, say, for digit representation of the hand, but when you start getting to more complex proprioceptive feedback, I think that there's a challenge there. It may not be impossible, but it's a challenge. So our approach was to take a very different perspective and to focus on the basic neuroscience of motor control, and to ask from that perspective, is there a new way that you could think about providing artificial feedback? And in particular, we're going to focus not so much on mimicking natural patterns of activity, but instead on taking advantage of the natural modes of learning and plasticity that we see that are really pervasive and quite important in sensory motor control. So now I'm going to switch gears a little bit and tell you about some of that basic science research that we've done in my lab. And I'm going to be building towards an explanation of why we're going to take what we call the learning-based approach to artificial feedback. So when you think about simple tasks like picking up a cup of water or orienting movements in space or whatever, there are really kind of, in my mind, at a computational level, 2 parts to that task. The first is the control part. So if I want to move my limb, I have to issue the motor commands, the efferent signals, that will go to the spinal cord, that will go to the muscle, that will make my limb move in the way that I want. Okay, so that's the control part. But before you get to the control part, there's another part. And that's what we call state estimation. That's the kind of engineering term for it. And what that means is that you have to know where your body is. You have to know the posture of your body. You have to know the velocity. And you have to know where objects are in the world around you. If you don't know that, then you can't issue the proper commands to get the movement to happen. So state estimation is really very important, both for the early part, the pre-movement planning part of motor control, and for processing online feedback, which we know plays an important role in volitional movement. Now we know a little bit about the kind of anatomy, the cortical anatomy that's involved in these tasks. We know, for example, that motor cortex drives movement of the limb. We know that premotor and various premotor cortices and areas along the interparietal sulcus are involved in the planning of those movements and are involved in the problem of state estimation, in particular along the interparietal sulcus of the macaque monkey here in parietal cortex. The IPS, this area, is critically important for that, for taking in sensory information and using that to estimate where the body is. And not only that, the IPS is a sort of a canonical multisensory area. It's not just taking in one sensory modality, but for example, in knowing where my limb is, I can use vision to see where my limb is, and I do that when I'm making normal movements. And then I also have proprioception to tell me where my limb is. So the IPS is constantly doing what we might want to call multisensory cue combination. That is, taking in multiple sensory cues and using that to inform me about some spatial variable, like where my limb is. Now, when I have my basic science hat on in my lab, the goal of the work that we do, the way I like to think of it, is to explain our computational understanding of state estimation. So there's been several decades of really good work in the psychophysical community modeling how it is that we use sensory information to guide movement, largely using statistical and optimal control frameworks. And I'll tell you a little bit about some of that. And what we want to do is take that understanding and try to map that to these neural circuits. And I'm going to tell you a little bit about the efforts that we've been doing in my lab to do that. And I'm going to focus again on this problem of multisensory cue combination, how you combine vision and proprioception, for example, to know where your limb is. Now, cue combination has become a fairly well-studied topic over the past decade or so. It's been studied in a variety of psychophysical settings, in monkey and in human, looking at visual proprioceptive, visual haptic, visual auditory, visual cue combination, all sorts of different modalities. And a dominant model has emerged, what I think of as a normative or predictive model, has emerged from that work, which is the model of minimum variance or maximum likelihood cue combination. So let me tell you what that means. What that means is that if you have some sensor, like vision, and it tells you about where your hand is, because of the fact that the neurons that encode the visual representation of space are variable, just because I see something doesn't mean that I know exactly, precisely, deterministically where it is. There's still some uncertainty in the representations in my brain about where something is. And we could think of that uncertainty as a variance, which I'll call sigma 1 squared. So this is the variance of, say, vision. And similarly, my proprioceptive sense has some variance or uncertainty associated with it. So this is just the amount of residual uncertainty that remains when I see something or when I feel something about where something is in the world. Now, because of this variability that's inherent in sensory and motor processing, because of that uncertainty, it means that if I have two sensory modalities, I can combine them. If you think about, if I want to know how tall I am and I take a measurement using a ruler, and then I take another measurement using a ruler, they're not going to be exactly the same. What's my best estimate? Well, my best estimate is to average them, right? And that's what's going on here. The best thing you can do if you have two sensory modalities is to average them. So you take one modality, you take the other, and you average them. But you average them, each one is weighted by the reliability of its cue, or 1 over the variance of its cue. So for example, if the first sensory modality is much more reliable, that means that sigma 1 is much smaller. That means that 1 over sigma 1 is bigger, so I'll weight that modality more than the other modality. So this may look like a fairly complicated sort of thing, but all it really says is that you take your sensors in, you average them, but you weight them according to the reliability. And if you do that, then the combined variance of that estimate is this, and this expression here, and that will be lower than the variance of either of the estimates alone. So the point here is that by combining two things, I reduce my variance. It's a good thing to combine two cues because it makes my performance better. That's the point. And not only that, we know the right way to combine them, and that's this. Now that can be put into a statistical perspective. So if I have an uncertainty about one cue, I can think of that as a distribution. Given my current sensory stimulus right now, this is my uncertainty. This is the distribution of where I think my limb really is. And the same thing for Q2. And when I combine them, I get an answer that's in between and that has a smaller variance. It's a narrower peak than either of the two, than Q1 or Q2 alone. So this is the statistical version, a probabilistic representation of the same thing. Okay, so in any case, we know that to a first approximation, this is what people and monkeys do when you put them in the context of trying to combine two cues about where something is in space or where my limb is in space. And there are even some network models for how this might be implemented in the brain. Work by, for example, Alex Pouget, Peter Latham, and researchers that have worked with them. But these models they're sort of hand-wired. They work with simple representations, and they work by kind of setting up the right kinds of network weights that would give you the right computation. There's even some very preliminary evidence that maybe properties like this are at play in the circuits involved, in cue combination in sensory cortices in monkey. But again, just very early evidence for that. So these models, again, although that sensory work is really looking just in a very sensory context of taking two cues in and asking, say, for example, am I moving to the left or to the right? That's work by Dora Angelaki and colleagues. So, okay, so there's this nice model of how sensory and how cue combination works. And so we can ask, is that how it works when you're thinking about state estimation for motor control? And the answer is no. That simple model that I've just told you doesn't seem to work in that context. Why? Well, we had shown years ago that when you look at cue combination for motor control, things look more complicated. Why do they look more complicated? Well, first of all, the weighting that you see between vision and proprioception seems to depend on things that it shouldn't depend on. So remember I just told you the only thing that should determine how you weight vision and proprioception is the reliability of those two cues. So for example, how I weight vision and proprioception of knowing where my arm is shouldn't depend on whether I'm reaching to a visual target or whether I'm reaching to my other hand, for example. But it does. So that's an oddness that wasn't predicted by the theory, but that we had observed. Furthermore, we showed that there are different computations involved in reach planning. For example, deciding, do I want to move to the left or do I want to move to the right? And then given that, how do I execute the proper motor control commands to make that happen? Those are different computations, and they seem to rely differently on vision and proprioception. The weighting was different. And neither of those would seem to be predicted by the simple model that I just told you about before. Similarly, in terms of the physiology, we've shown that if you look at the weighting of vision and proprioception across different brain areas, it seems to be different. So if you look at two different brain areas within the parietal cortex, or if you look at parietal cortex versus premotor cortex, the weighting of vision and proprioception is different, which again, if there was some simple notion of this is the visual reliability and this is the proprioceptive reliability, that's not what you'd expect to see. So does that mean that state estimation for motor control is not optimal? Because I told you that sort of weighting is the right thing to do. It's the statistically optimal thing to do. Does that mean that our movements are more variable than they need to be? Well, maybe not. And here's why. I showed you this simple picture that says, vision and proprioception come in to the interparietal sulcus, or the posterior parietal cortex, and they're combined. So A, B, two modalities come into a combined area, and you combine them statistically optimally. So that's a nice picture. But that's not really what's going on. What's really going on is there's a complex network of brain areas that are involved in state estimation and motor planning. Many of these lying in the posterior parietal cortex and along the interparietal sulcus. So there are at least a half a dozen areas involved in state estimation for reaching just along the IPS. So that's more complicated because what it means is that these brain areas aren't really integrating vision and proprioception in some canonical sense. What this particular brain area here, for example, does is it simply integrates its own inputs, its afferent inputs. But those inputs come from other brain areas. So there's sort of a cascade. There's already processing that's been going on before you get to the brain area of interest. And one example of that is the computation of coordinate transformations. So for example, I talked about taking vision and taking proprioception and combining them. But that's not really quite the right way to think about it, because if my hand is over here, then proprioception is telling me where my hand is in some body-centered space. It's saying my hand is here with respect to my body. But when I see my hand, say over here on my retina, that's dependent on where I'm looking. So if I shift my gaze to the left or I shift the gaze to the right, then the location of my hand on my retina changes, but of course my proprioceptive sense doesn't change. So in order to map the visual retinotopic location, this red arrow, to where I feel my body to be, the blue dot, I need to know where my gaze is. And that requires a transformation. In this simple vector notation here, you have to add the retinotopic vector to the gaze location, and then I get where my body is in space, where my arm is in space with respect to my body. Right. So the details here don't matter. The point is simply that to combine two sensory modalities, you have to add other information. You have to do some processing. And it turns out that kind of vector addition, it can also be cast in a probabilistic terms. And in this case, it says that if you know where your body is, if you know where you see something to be, and you know where you're looking, you can compute where something should be with respect to your body. So you can take these two things and add them, which winds up being a convolution instead of a multiplication. The probabilistic computations don't really matter. It's just to say that when you do this, you get a different variance. And in this case, the variance that you get, where you predict the hand to be with respect to the body, after combining visual and gaze information, is bigger. So it increases my variance. So again, the point simply is that if you want to combine information from a bunch of different sources, that requires doing some processing on that information. And typically that will involve changing the uncertainty associated with that variable, often increasing it. OK. So that means that maybe these observations that we've made, that the weighting of vision and proprioception seems to be different in different brain areas and seems to be different for different computations and so on, maybe that can be cast in a really simple engineering framework that makes a lot of sense, which is I have different inputs like vision, and that's going to come in an eye-centered representation because when I move my eyes, vision moves with it. That's fairly straightforward. And I have inputs like tactile inputs or on my hand, and those are going to come in a hand-centered representation because if I'm tapping my one finger against the other, that percept doesn't change as I move my limb around in space. So that's locked to the hand. And if I want to relate those two, then I have to use information about my eye position and my head position and my wrist position. So you can imagine there's a series of representations, each in a different frame of reference with respect to the body, and there are simple transformations back and forth. Maybe this is what's going on in the brain. Nice, simple engineering framework that was proposed by many people back in the 70s and 80s, for example. But it turns out that it's not right. And I'll tell you why this simple kind of linear model of transformations isn't right. It's that if you go into the brain, you never see simple things that look like this. You don't see representations that look like an eye-centered representation or a body-centered representation. So I want to tell you just a little bit of physiology here. I hope it's not, I hope I don't blow through it too quickly. But here's the idea. You can record from a neuron, and you can ask, how does the pattern of activity that you see from that cell, how does it relate to these representations? So for example, you could have a cell that when I move my eyes, the visual world moves with it. And that would be a retinotopic cell. Or you could have a cell that when I move my eyes, nothing happens. So for example, it encodes a target over here. It fires a lot when I'm going to reach over here, but not over here. And it doesn't matter where my eyes are. So that means it's encoding a target location with respect to my body. But if it's encoding a target representation with respect to where I'm looking, that's a retinotopic representation. So you could have either one of those in the brain. If it looked like that, if it was a retinotopic representation, we'd say it looked like it was over here. If it was body, we would say over here. Now we can generalize that to a form that looks like this, where the cell is encoding where something is with respect to my body, plus some multiple times where I move my eyes. If that multiple is 1, then it's a retinotopic location. And if it's 0, it's a body-centered representation. Again, the details here aren't that important. It's just to say that we can quantify the pattern of activity of how cells encode things in space as being either more like a retinotopic representation, delta equals 1, or more like a body-centered representation, delta equals 0. And if you go into areas along the interparietal sulcus, like area 5 or MIP, or from Richard Anderson's lab, this is work from my lab, from Richard Anderson's lab, an area more medial to these two areas, which he calls the PRR, or parietal reach region. What you see is that these representations are kind of all over the map. He uses a different terminology, but it's the same idea. You have some cells that look like they are retinotopic and some cells that look like they're body-centered, and in fact, some cells that don't make any sense at all. They're less than 0 or greater than 1. It's a mishmash. It's a complex set of representations that don't seem to obey anything like that simple linear model that I was telling you about. Not only that, you have different brain areas that all sort of overlap. This one, if you take the mean here, it's a little more like a body-centered, and this one's a little bit closer to 0, this is a little closer to 1, and so on. So there are subtle differences between these areas, but they're highly overlapping. So the representations are complex and they're redundant. Now, stepping back from the physiology, I told you at the beginning that our goal was to explain the computational element of things like we combine information in an optimal way, we use that to guide movement, to explain that in terms of neural circuits. But that's complicated by the fact that we have these complex, heterogeneous, and redundant patterns of activity. Now, it's not even clear how that works in the brain, because if you have these complex things, how does the brain even know how does it know how to make sense of them? And the answer appears to be learning. So the mapping between one modality and another is highly plastic in adults. Say, for example, prism adaptation is a good example of that. If I put prisms on, very quickly my brain readapts to bring vision and proprioception back into alignment. Not only that, there's good evidence that the ability simply to use vision and proprioception together to combine those signals or any intersensory coordination seems to require learning during development as well. And so in thinking about how that works, when you've got these complex representations, how is it that the brain learns to combine those sorts of things? And we don't know, of course. That's a really great, I think, one of the deep questions about sensory processing, unresolved questions about sensory processing in the brain, and how we learn to do these complex things during development and how we relearn as an adult. But in this case, I think theory can be a catalyst. So what I'm going to do now is going to spend about 5 or 10 minutes telling you about some recent theoretical work we've done. And at the end, I hope that'll give you an idea of the way that I think about this problem, and it'll also point the way toward going back to this problem of artificial feedback. Okay, so this is work that was done by Joe Macon and Matt Fellows. Matt left my lab, but Joe is still in my lab, a really great theorist who's actually on the market now. So if you guys are hiring and know anyone who's hiring a theoretical neuroscientist, he's really fantastic. What our approach here was to start from the perspective of machine learning. So imagine the following. Imagine that you've got two sets of neural representations. This is a network model, okay? So we've got this grid here is meant to represent a bunch of neurons that represent, say, proprioceptive or visual space. So each point on the grid is a neuron, and so if my arm is at some point in space, that means that some theta 1, theta 2, some joint angle position, then these neurons here, say, for example, will be firing more, so I've colored them in. And that maps to some set of neurons over here that correspond to the visual location of where my limb is over here, and they'll be firing. But of course, there's a nonlinear mapping between where the visual world is and joint angles, and that's supposed to be represented by the fact that this rectangle here maps to this kind of moon-like shape over here. So there's this kind of complex scheme of representation and a nonlinear mapping between them. And the question is, how could the brain learn to integrate these two things, to combine these two cues? So our idea was that maybe we shouldn't think about the problem as the way I drew it before, x1 weighted, x2 weighted, take the average. Don't think about it in terms of that simple mathematical framework, but instead think about it from an information processing framework. What the brain's going to get is a bunch of information, a stream of visual information and a stream of proprioceptive information. And it wants to find the things that relate the two so that it can learn how to combine them. In other words, it wants to take all that stuff, all the noise, all the stuff that doesn't matter, winnow that out, and find the common things that agree between the two signals. And that idea of winnowing out the information that's unimportant and learning about the information that is important, statistically speaking, is exactly what unsupervised learning algorithms in the machine learning world do. And in particular, there are lots of algorithms that what they do is they build efficient statistical representations of their inputs. So we're going to throw a network architecture and a learning algorithm at this problem. And I'm not going to tell you too much about the details because they're not that important, except to say that they're kind of a generic algorithm that comes out of machine learning. And the point of this algorithm is to just learn efficient representations of the statistics that you throw at it. And our idea was that if you do that, over time, this upper layer, the network will learn to create efficient representations, and therefore will learn to do Cue combination. That was our idea. OK, so it turns out this works. So I'm going to show you one or two quick examples of that. So here's the output of the network in the following sense. This is a kind of a condensed representation. So you can take these neurons here, just these visual neurons, and you can ask, if I decode them trial by trial, what's my error in knowing where my limb is? And of course, it's a two-dimensional location, so that gives you an error covariance. So that's this purple ellipse. So that means that I'm not very good at estimating vision along this axis, but I'm pretty good along this axis. I'm OK. I've got a bunch of error. That's what I've got. What if I just look at the proprioceptive neurons? This is the best I could do. So that's the optimal decoding, which we can actually write down theoretically, you can derive what the optimal decoding of these two sets of neurons is. Because remember, these are model neurons. That means we use Poisson firing. In other words, with the same input, same location of my limb on every trial, I get different patterns of activity trial by trial in the neurons because we're simulating noisy neurons. And that means that there's trial by trial variability in the performance of the network. And then I could say, well, if I have those two inputs, I can just ask theoretically, what's the best I can do in combining them? And that's this black curve. Then we take the network and we train it up with these inputs trial by trial. And after learning, we ask, how well can you decode simply looking at these green neurons? So we don't have the original inputs anymore. We just have the green neurons. And it turns out it does optimally. It does just as well as you can expect, optimally combining those two inputs. And that's true. This is one location in the workspace, but that's true everywhere in the workspace. So the point here is that this network has essentially learned to perform this statistical operation. It's learned to do optimal Q combination, but it's learned it de novo just by looking at the statistics of the inputs. The network can do other things too. Remember I told you before that one brain area, you can think of it as only combining input from multiple brain areas. It's not really combining vision and proprioception. It's combining input from earlier areas. And the network actually can work in this kind of compositional way. So if you train the network, you take those green neurons, and you give a bunch of green neurons from one network into another network, and you give them to a fourth network, that network can also learn to encode those signals in an optimal way. So it kind of propagates forward in a statistically optimal way. The other thing that it can do is it can do that problem of computing sensory transformations. So if I give the network information about where my, let me skip that part. If I give the network information about where I'm looking, gaze information, and I give it information, eye information, I give it information about where my lib is in space, and I give it information about what I see, now in order to map what I see to what I feel, I need to know where my eyes are looking. So now the network has 3 inputs instead of 2. And when you do that, it essentially learns that coordinate transformation. So it gets that statistically right as well. So I went through that quickly, but what I'm trying to tell you is that what we found, actually much to our surprise, is that the network is able to learn all the sorts of things that we know parietal cortex does during state estimation. It learns Q combination, it learns coordinate transformations, it learns something called Bayesian integration, which is that if you have more data in one part of the workspace, it learns to weight that more. It does all of the things that we've seen psychophysically, but it does it without having to be told in advance what it's supposed to do. It just learns it from the statistics of the inputs. And not only that, it can also learn, I've told you about static networks, but it can also learn dynamic networks. So if we have information that varies over time in a predictable way because, for example, you're looking at your real limb, it learns that as well. OK. So what's my point? The summary of all that theoretical work is that, so let me tell you one more thing, which is that if you look at the hidden layers of that network, this is the network activity, you get, again, a mishmash, a mix of representations. Say, for example, in that case where I have eye information, hand information, and visual information, I get this complex mix of representation that looks a lot like the data that Richard Anderson collected. So again, in this network, you get these kind of complex, mixed, heterogeneous representations, including some cells that don't even make sense, being kind of off on the edges of not being retinotopic or body-centered. So again, this sort of looks like what you would expect to see in the brain, these kind of complex things. All right, so what's the point? The point is that maybe the way to think about parietal cortex is not in terms of doing x1 plus x2 weighted or something like that, but the way to think about parietal cortex is in terms of learning. That the job of parietal cortex is to learn to make sense of the data that it's given. And we've shown that if you do that, if you get a simple network model to do that, that network model can learn to do all the simple computations that we think are going on in state estimation for movement planning. And this model is consistent with the complexity we observe in terms of that we don't expect simple representations, we don't see them in the network, and that if you compose a bunch of these networks in a hierarchical fashion, nearby networks will contain similar but not identical patterns of activation. So how can we test something like this? Well, the proposal here is that things should be complicated. That's hard to test. But the other proposal is that, that representations and behavior should depend on the statistics of signals in a predictable way. That's what comes out of the network model. And that the changes in representations during learning should also depend in a predictable way based on the statistics. So it's not clear yet exactly how you would do these experiments, but imagine recording from multiple areas within the parietal cortex while you're looking at complex problems of state estimation and while you're looking at learning problems. And somehow you might be able to map what you predict to see in the model to the network patterns of activity. Now, at the end, I'll come back to this briefly because I think that the manipulation I'm going to tell you about for artificial feedback might give us exactly the tools that we need to do this. So I'll come back to that. OK, but now finally, let me say that this theory, again, points to a way to go to achieving artificial feedback. And what do I mean by that? The first thing I said about the model was that the statistics determine the computation. That's what we learned. You just give a bunch of inputs, and the network learns to do kind of the right computation. And in particular, if you give the network two inputs that correlate moment by moment, trial by trial, vision and proprioception, say, the network learns to do optimal cue combination. So that ability to learn to correlate, if it's true that that's what the brain does, then that suggests a way to give artificial feedback. So how? Here was our idea. This is work that was principally done by Maria Dotterlot, a graduate student in the lab who's now a postdoc, and Joey Doherty helped as well, and he's following up now with the sort of more straightforward BMI application. So what I'm about to tell you is an experiment that isn't really providing artificial somatic sensation to a BMI. It's a little bit more of a complex experimental design. But at the end, I'll tell you that we think it translates directly to the problem of artificial somatic sensation. So again, the question that we're asking is signal correlation, if we give the brain something novel, but it correlates moment by moment with something the brain knows about, is that enough to drive integration of that signal, to learn to use that signal? So here's our experiment. We have a monkey that's been trained to make reaching movements from point A to point B. This is standard fare for monkey reaching experiments, very boring stuff, reach here, reach here, reach here, and so on. The difference is that instead of letting the monkey see where that target is, we're going to give the animal a different kind of cue. We're going to let him see a bunch of dots, randomly located dots, moving on the screen. I'll show you an example of that in a few minutes. And if those dots are moving, say, outward and to the left, or outward to the right, that means that the monkey knows he has to move in that direction, outward and to the right. And if the dots are moving fast, that means that the monkey knows he needs to move far. And if the dots are moving slow, that means that he knows he needs to not move very far. So this is just something that he's learned to do. And I'll show you, again, some examples of that. The nice thing about this experimental design is it's very easy to control how much information there is about the direction of the cue. If all of the dots are moving together in the same direction, it's a very salient percept that there's flow in that direction, and the monkey will be able to perceive it with little uncertainty, with high reliability. On the other hand, if, say, half the dots are moving in a random direction and half are moving coherently, that's what we call 50% coherence in one direction, then you can still see it, but it's harder. You have to pay a little more attention, and you're a little more uncertain. And we can go all the way down to zero coherence, where there's no directional information at all in the cue. OK, so that's the baseline task. We train them to do that. And then what we do is we start stimulating in somatosensory cortex, S1 of the monkey. And we do this in a pattern, which I'll tell you about in more detail in a minute. But it's a completely non-biomimetic pattern. So this is something the monkey will have never experienced before. But moment by moment, it's going to correlate with this visual cue. And our hypothesis was, based on that theoretical modeling that I was telling you about, when the parietal cortex, when the brain gets, moment by moment, over and over again, things that are telling it about the same thing, what it does naturally, effortlessly, is it learns to combine those two things. It learns to integrate them. That's the idea. And then, of course, once that's done, we can test performance with that ICMS, intracortical microstimulation. ICMS is the term I'm going to use. We can test it with that stimulation or ICMS signal alone. OK, so this is what the task looks like. Here's the monkey. He's going to be reaching to an unseen target over here. So first he goes to the start position. The dots come up, and now he's going to reach. And what you can see is the monkey's pretty good at this. This is not a trivial task. He's correcting online, and that feedback changes online as he moves. So this is a signal that is not only providing a cue at the beginning of the movement, he has to wait for a go cue. So he gets this flow, he can look at it, he can think about it, then the go cue comes, and then he can start to move. So this flow is telling him it's a pre-movement cue about where the target is, and then when he starts to move, it's a feedback signal that he can use to correct online. Okay? All right, so that's the baseline task. It's a little challenging, but he gets good at it. Any questions about the task? Yeah.
So do the direction of the dots change depending on the monkey's position?
Yeah. So what these dots are always encoding is the relative position between where the hand is now and where the target's going to be. So for example, if the flow were outward and I overshoot, now the flow is going to be inward. Okay? And if I overshoot only by a little bit, the flow will be inward but slow. Okay. And of course, the same information is going to be encoded by my ICMS signal when I get there, and it will also be updating moment by moment online. All right, so we can quantify performance here in a variety of ways, but I'm just going to tell you about one, and that's the variability of the initial angle of the movement. So if you knew exactly what to do, and you were trying to move efficiently, you could just move directly to the target. But monkeys don't do that. This is an actual trajectory from some movement. He goes over here, and then he corrects. And we can look at that initial angle, and then we can look at how variable that initial angle is. We're not going to care yet about the bias, although I'll come back to that at the end. Right now, we're just going to look at the variability of that initial angle. And of course, what you see is that when, remember, just the visual task, when vision is highly reliable, because the coherence is low, this is performance for two different monkeys, the variability of that initial angle is quite low. And then as the coherence drops, so the signal becomes noisier and noisier, and it's harder and harder to estimate which direction you should be moving, the variability of that initial angle grows, as you would predict. Okay? So this is just the baseline psychophysics. All right, now, again, and this shows that we can modulate the reliability of that cue with the coherence of the dots. All right, now what we're going to do is we're going to do cortical stimulation. So how are we going to do that? We have an array of 100 electrodes that sits in primary somatosensory cortex of the monkey. And we're going to select 8 electrodes on that array that we know the monkey can feel or can detect stimulation on. We do that in a separate task where we just ask him, do you or do you not feel stimulation? We find 8 on which he can, and then we go forward. Now we assign each of those 8 electrodes in an arbitrary way a preferred direction of movement. And again, this is arbitrary. So for example, if the target is off to the right, this E1 electrode would be stimulating with a fixed amplitude biphasic pulse, would be stimulating at the highest frequency. So all we're going to change is the frequency of stimulation, not the amplitude, not the shape. Similarly, as the monkey gets closer to when the monkey's far from the target, all of the electrodes are going to be stimulating at a higher rate. And as we get closer to the target, that average stimulation rate will drop. So again, the average stimulation rate across all electrodes encodes how far the target is, just like the speed of the dots. And the relative distribution of firing rates across these electrodes determines which direction the monkey should be moving in, just like the direction of the flow in the dot field. Any questions about that? OK, so this is the setup. So here's an example. The monkey originally needs to be moving, say, around 150 degrees maybe. So these 2 electrodes are pulsing at the highest rate. He starts to move. He overshoots a little bit. So now the electrodes encoding the opposite direction are now stimulating at the highest rate, and then he achieves the target. OK, note this is a non-biomimetic signal. The monkey's never experienced anything like this before. So we train the animal on this. Now it takes a while, but this is what a training trial looks like. The monkey sees a bunch of optical flow, and he was receiving these dots, and he does the task. Now, of course, he can already do the task with vision. So you might think that look, he can do the test with vision. There's no reason for him to pay attention to this artificial stimulus. It's just a nuisance. If he ignores it, he can do the test just fine. But our argument was that what the brain is always doing is assessing statistics of inputs and trying to do the best thing it can, the right thing with those inputs. And under that argument, because these correlate moment by moment, the monkey shouldn't ignore these cues because it's providing it valuable information that could allow it to improve its performance. And sure enough, it took a while, after weeks, the monkey got to the point where he could do the task with the ICMS signal alone. So now the monkey's sitting in a dark room. Of course, he doesn't see that target. That's just shown so that you can see where it is. And he's able to make that movement to this target, albeit a little bit more slowly. But he's able to make a movement to this target just guided by what he feels in somatosensory cortex from that pattern of stimulation. OK, so let's quantify that. This is the psychophysics of the visual performance that I showed you before. And this is his performance with ICMS signal alone. Now, if you look at this, what this means is that he's performing as well as he would with 25 to around 25% coherence. Now, I have a demo up on my laptop. So afterward, if anybody wants to come up and see, I can show you what it looks like to have 20 to 25% performance, 25% coherence. It is some challenge, but you can definitely see it. And these monkeys are fairly well trained, so it's actually pretty easy for them. So he has a pretty strong percept here. They have strong percepts when they're making these movements just from the stimulation. OK, now that's interesting. But of course, the argument I was making is that the reason to combine two cues is because performance is better. So the prediction is that if he has both cues, not just ICMS, he should do even better. And this is the performance we see in red when he has both cues. Now, if you look over here at the right side of these plots, that's the case where vision is really good. And notice there's not much difference between blue vision and red combined vision and ICMS, because vision's already really good, not surprising. Now, if you look over here on this side of the plot at 0% coherence, that's the case where vision is terrible. It basically gives you no information. And you can see that you certainly don't get any improvement. In fact, you might do a little bit worse, and I can talk about that later if you want, but you don't do better. But the prediction, remember I said, was that you should do better. Well, you should do better when it makes sense to combine them. Where does it make sense to combine them? At the places where they're sort of roughly on par with each other in terms of reliability. And indeed, in the middle of the plot, the red is better than the blue or the green for both monkeys, and that's significantly true. Well, OK, but I told you that we had this nice theory about how cues should be combined. So is the result consistent with that theory? So what does that mean? That means that the two cues should be combined in that statistically optimal fashion. And remember, this is the equation I showed you at the beginning that says that the variance should decrease by some predictable amount, and that the average of the two, we could compute what that should be too. So we could compute the variance of the behavior and the mean of the behavior that we would expect to see if the monkey were combining optimally. So what I'm going to do first is I'm going to show you, indeed, that we get this improvement consistent with what we'd expect to see. And then I'm going to go back to this mean argument as well at the end. So the red data here are what you see with the combined cues, and the dashed red lines are what theory predicts. So again, what we're seeing is that the animal is able to combine these two cues, and it appears that he's combining them in an optimal way to get the best performance he could, given the two individual cues, which is exactly what an animal would do with two natural sensory cues. So not only is he doing great, but he's treating this signal just like he would for natural signals, combining it with his natural vision in a natural way. Now you might say, well, yeah, whatever, Sabes, that's all right, but I mean, come on, there's a floor effect here. That's not so impressive. Well, we can analyze the data in another way here, which is that it just so happens that there's some natural bias in performance that's different between the two cues. So this monkey here, for example, if you look at just the direction of the reach, should it be reaching over here, over here, over here? This is sorry, that says visual coherence. I apologize. That should say angle of reach. The angle of the reach here, and you look at the mean initial angle. Now, we're not looking at the variance, we're looking at the mean. That mean initial angle changes across the circle with vision. And it changes differently with ICMS. Why? I don't know. It just does. But then you can say, what happens when you have two cues? Well, with 15% coherence, what happens is that the red curve lies largely in between the green and the blue. In other words, it looked like he's doing something in between the two, which is what you would expect if he were averaging. So those data can be sort of put in this kind of normalized framework where 0 means that you're lying just on top of the green line, blue means you're lying just on top of the blue line, and he's somewhere in between with 15% coherence. Then we could ask how he does with other coherences. Well, at 100% coherence, when vision's really good, the data look a lot more like the visual bias. At 0% coherence, when vision is terrible, and he should be relying on ICMS, the data look a lot more like the ICMS bias. And indeed, the data look an awful lot like what we would predict from the model. So in summary, what we've shown is that this learning-based approach seems to work from an arbitrary mapping of variables of ICMS, so something entirely novel that he's never experienced before. The animal is able to learn to use that cue in a statistically optimal way, driven by the correlation between the two cues. And then at the end, he's able to use it in this natural way, combining it with vision. And we think that this result, which we're very excited about, we think it has interesting implications both for BMI and for basic neuroscience. We briefly go back to this argument about the complex computations and representations we see in this area. We think that with this technique, it gives us a really unique tool for studying these circuits. We can go in and we can directly manipulate the activity patterns here. We can create novel signals. We can watch how they get integrated into these circuits. We can change the statistics of them in a way that we control. So we think this is going to give us a novel tool to start comparing the complex physiology that you see in parietal cortex to the models. In terms of BMI, of course, I think it's fair to say that this really sets a new benchmark for ICMS delivery of in the brain, cortical stimulation delivery of write-in of sensory information. Multi-channel, multivariate, real-time feedback. I didn't tell you about about this, it's in the paper, but we can show not only is he using that signal to plan the movement, but he's using it to correct online. So this is, I think, a new benchmark. And we also think that it generalizes in a very straightforward way to artificial proprioception in the context of a real BMI. So Joey O'Doherty, the postdoc in my lab, is doing this work right now, where a monkey's controlling a cursor on a screen, and we hope that soon that monkey will be able to feel the location of the cursor through this stimulation. Now, I'm done, but I just want to say two more little remarks that relate this to the kinds of work that's going on here. And the first is the relationship between learning and biomimetic approaches. So at the beginning, I said that there are these people who think about biomimetic approaches, and it works for the cochlea, but it'll never work for proprioception. So you should do this new thing that I'm telling you about. But that's not actually the right way to think about it. We can never truly achieve biomimetic stimulation. Even the cochlear implant isn't really biomimetic. When you first put a cochlear implant on someone, it doesn't sound so great. They need to learn. They need to get rehabilitation before it starts sounding good. And in the brain, there's no hope that we'll ever recreate truly natural patterns of activity. And so I think that biomimicry is actually a continuum. There are things that look more biomimetic, and there are things that look less biomimetic. And the question is, how much is it worth doing research and engineering and pushing and spending time and dollars to get more and more and more biomimetic when the brain ultimately can cover a lot of that gap? That's the argument that I'm making. And the way I think about this actually is through this curve here. That over time, you can think about the performance of a neuroprosthetic device. So there's the performance that you get day one out of the box, initial performance, first trial performance. There's the asymptotic performance after lots of learning, how well can you do? And then there's the learning rate. How long does it take? How much rehab do you need to get to that point? Now clearly, the more biomimetic you are, the better you're going to do out of the box. If you start with something the animal's never experienced before, he's not going to do well out of the box. But it's my hypothesis that the learning rate and the asymptotic performance won't be so affected by adding finer and finer layers of biomimicry. That getting close enough is probably good enough, and that with a little bit of rehab, or maybe a lot of rehab, you'll be able to get great natural performance. That's the hypothesis. Remains to be seen. And I want to say one more brief thing about the relationship between central and peripheral interfaces. As many of you know, Dustin has done some work that's very closely related to this, providing both motor outflow and sensory feedback in the context of peripheral devices. And I don't think that there's a right answer here. It depends on the population, obviously. Peripheral interfaces are less invasive, and they make use of the residual neural circuitry, the spinal cord, et cetera, and thalamus, and so on. That's a good thing, presumably. But on the other hand, Central interfaces, I think, ultimately might allow for much greater bandwidth and greater specificity in the responses. I like to think about it as that what the brain is there for is to be a breakout box for the neural engineer. Dustin is trying to interface with the Ethernet cable. I'm walking over to the breakout box with the junctions in it. So, maybe there's an advantage to that in the long term. And so I think there may not be a clear right path, even in the case of amputees. Ultimately, there may be some patients for whom it makes sense to have a peripheral implant, and some patients for, even in the case of amputee, it might make sense to have a central implant. And at that, I'll stop. Thank you.
First, we thank you for coming.
Thanks.
And so it turns out that Flip has connections to this area and actually spends a lot of summer in Vermilion at a home. So as a token of our appreciation, we'd like to give we have a book here, taken from a local person with photography from many of the landmarks and stuff in Cleveland. So you take this back to your wife and say,
Thank you. That's very nice.
Connection to the area.
And I have a standing invitation. Just send me an email, and this summer, you guys can come swim out in Vermilion. I've got very nice in-laws. They'll be happy to host. You're all welcome.
So, yeah, go ahead, Paul.
Sure. Okay. This is very cool and beautiful stuff. I have a question about this. I mean, it appears that it works like gangbusters immediately, and when you go in and you're doing this cortically, you're essentially operating in a substitution sort of framework where you're substituting one type of input for another type of input. And when you go out in the periphery, typically the substitution approaches have been demonstrated to actually sort of inhibit they're considered much more of a distraction and will actually decrease performance when you're getting exactly the opposite effect here. Mm-hmm. And so what do you think the difference between the periphery and the central sort of input is?
Yeah, that's a great question. And let me just say that I didn't really talk about, obviously I tried to put a fair amount into the talk, so I didn't have time to say everything, but this work was heavily motivated by the kind of sensory substitution approaches that people have done. And there's successes and failures of that approach. In some cases, it's really beautiful. You see changes in percept with experience. But on the other hand, the performance is never really quite as good as you want. And indeed, it can even become a distraction. Now, my answer to that is twofold. So first, let me answer the question you didn't ask, which is, if you're going to do this, and you're doing sensory substitution anyway, why bother opening up the head? Why not put a tactor array on the back or whatever? And the answer to that is that what we want to do here is to create high fidelity, high bandwidth, low lag feedback. And I think there you will have much better ability to write in high bandwidth signals directly into the brain than we will be able to do with a tactor array, say, for example. And ultimately, I think that when the neural interface devices that we in my own lab, I didn't talk about it, but we've been developing what I think is some very interesting novel devices using what I think would be minimally invasive, very large scale interfaces with the brain. As these next generation and next generation devices become available, where they can become fully implanted, wireless, high fidelity, robust, then it would be much better anyway to have something like that than to have this big backpack that you have to carry around with a tactor array. So anyway, that's the question you didn't ask. But then why is it that we see such a great result when sensory substitution can sometimes be a distractor, can actually cause a decrement in performance? Well, I didn't show you, let me see if I have, I can't remember if I have this plot or not. I didn't show you the learning curves. They're in the, unfortunately I didn't put them in my talk, but they're in the paper. If you look at the actual curves over time, performance over time, what you see is at the very beginning when we start stimulating actually does cause a decrement in performance. So stimulating ICMS plus vision compared to vision alone, you're worse with ICMS. And that persists for about 2 to 3 days, or maybe a week. So for a brief amount of time, it's worse. And then you go through a period of time where it makes no difference whatsoever in performance. And then you get better. And if you look at it, what it really looks like is a fairly smooth curve coming down. And so it is true that at first, it is probably disrupting normal proprioception, and therefore it's a nuisance. Or it's just a distraction. But again, it takes time. So I'm not arguing that this ability for the brain to merge is something that happens very quickly. We think we might be tapping into some of the same mechanisms that are involved in development. It seems it takes weeks. But over weeks, then you get to that point where you get the improved performance. So that's what I think is going on.
So just out of curiosity, so there's very distinct connections between V1 and S1. And for instance, if you're doing grading orientation thresholds and you ping the visual cortex with TMS, you lose the ability to actually discriminate direction. Is there a possibility that you're integrating or co-opting with that particular system so that you're actually integrating it as a visual system, as a visual input versus a somatosensory input?
Well, yeah, so I think that sort of how you're integrating it and is I think we're very interested in that. So this is something that we've submitted a grant on. Actually, that grant didn't do particularly well, but we'll try again. And I think for me that you can address this a little bit behaviorally, but physiologically is really the way to go. But what is it that's being learned? So you might imagine that what's being learned is something for reaching. That the monkey's got a lot of experience with these combined signals in this task. He's just learning something about this task. So one question that's a really interesting one is, does the knowledge generalize beyond reaching? So if the signals sort of getting combined in an early sensory area, then you might imagine that what you would see is that you could use the same signal now to improve performance on a saccade task, where you had to, like a Newsom or Shadlin task, where dots are moving, you have to decide whether to go left or right, for example. We'd like to do those experiments. We haven't done them yet. Similarly, if you go and you look at the parietal representations of these, of this signal, is it the case that you only see a merged representation or some kind of combined representation in the brain areas that were responsible for the task, say the reach areas? Or do you see them all along the IPS, say even in the areas that would be involved in shaping of the hand for grasp or in saccades? Or is it none of the above? Is it that representation comes in at an earlier level, more thinking is sort of merging along into the visual pathway or the somatosensory pathway? We don't know yet. So I think that it's a combination of psychophysical studies of the kind that people, that my lab and others do, of kind of parsing out the role that signals play in movement and just looking at the physiology. But this is a great question. How much is it task dependent? How much is it really a perceptual effect versus a motor effect? These are things that we're very interested in but don't know yet. Oh, sorry.
Go ahead, Pat.
Actually, my question kind of segues from what you just said there. What if the array was in M1 instead of S1? Would it still work? I mean, do you have any idea what the monkey perceives when the stimulation is going on?
Boy, would I love to know what the monkey perceives. This is a hard test to replicate psychophysically. Here's what I think. I think that you could train the monkey to do this task. Let me put this learning slide back up. I think that you could train the monkey to do this task with stimulation anywhere in the brain. But if you pick something, someplace pretty far, anywhere that he can detect stimulation. If you pick someplace that's not involved in the circuit at all, I think what you'll see is that it's going to take a really long time to learn, maybe well beyond our capability of following, and the asymptotic performance won't be as good. I think there's something about tapping into, let me not address M1 yet, but there's something about tapping into S1 that's special because somatosensory cortex has a tight loop with motor cortex. And that, and what we're looking for ultimately in the BMI context is providing something that's akin to proprioception that allows for the kind of rapid, tight coordination of feedback and movement, like the kinds of quick loops that Steve Scott's been documenting recently in the intact animal. That's what we want to recover, right? And so that's why we want to stimulate in S1, not in, MT or something like that. Now, M1's a different story, of course, because that's the motor cortex. There, you would think that it would be something more like a command or a goal, or PMD, dorsal premotor cortex, same sort of thing. I do think that the animal could learn that. What I don't know is whether it would be combined in an optimal way, and it's a really interesting question. Something that's sort of, I don't know.
But there's a handful of people that have M1 arrays, is what I'm trying to get to.
We have M1 arrays. We could do it. We're doing an experiment right now with an animal that has an M1 and an S1 array. We're not doing that, though, of course. We're not doing that experiment because...
You could ask the person what they would perceive.
Oh, people.
Yes, there are people.
Yeah, sorry. There are people.
So there's M1 arrays.
There are people. Okay,
Oh,
okay.
I
So
don't know that.
there are people that have M1 arrays.
Yeah.
There either are or very soon be people with S1 arrays. I think there are. Is that correct? You know, Pittsburgh. Anyway, there will soon be if there aren't already. As far as I know, and you would know it better than I, but as far as I know, no one has FDA approval to do this kind of stimulation in those M1 arrays. I could be wrong about that.
I don't know. So
Okay.
I don't know.
I would love to try something like that. I know that the Pittsburgh group is close to doing stimulation in S1. I think that they're going to try a more standard approach at the beginning. I have offered them a standing invitation to get involved if they decide that this sort of approach might be better. But I would be extremely eager to collaborate with anybody who has access to humans where we could try these things out. I do think, these were long experiments. This is months. But I do think that there are versions of this that we could do in a day or in 2 days that I think would provide some really interesting insight. And it's very exciting. It's something that we've talked about. I talked about a little bit with colleagues at UCSF, with ECOG. Stimulating with ECOG isn't quite the same as stimulating through a penetrating array. We're eager to do it. We haven't been able to manage to get it yet, but I'd love to do that.
Well, I think you partially answered my question because a lot of the clinical applications, for example, a patient with a stroke, I mean, this learning, does it require intact cortex or can you retrain? I mean, how plastic is the cortex if you've got a frontoparietal infarct from a middle cerebral occlusion?
Right.
How do you select, theoretically, how would you select what area in a human with that kind of stroke to stimulate?
Yeah, that's a great question. So a postdoc in my lab, Azadeh Yazdan Shamurad, is very interested in exactly this question. She actually has a AHA fellowship to look at this issue. We could have started with a stroke model, but obviously that's a little more complicated in the monkey. So what she's doing right now, it doesn't quite get at your question, but we're doing something that's very different from what I told you about, which is that we have a cylinder on a chamber on the animal that's exposed a very large portion of M1 and S1. So this is an inch diameter cylinder on monkeys, so it's a pretty good amount of tissue. And we've, using a trick that we got from a colleague, Chris Bankowitz, and his collaborators at UCSF, we've got optogenetic expression of C1V1, one of the optogenetic channels, throughout almost that entire region of cortex. So now if we shine a light almost anywhere there, we can activate tissue. And then in collaboration with Michel Maharbis at Berkeley, we've got these arrays, these ECOG arrays that we can lay on top of the tissue, stimulate and record at the same time. So we have the capability of stimulating and recording and looking at, now not the fine scale neuron, but kind of the map activation patterns. And what we're doing now with that is exactly this kind of idea of testing, what kinds of local plasticity rules can you get? If you knew that something was here, could you create a jumper cable in essence over? And we're starting to play with those things right now. It's very early days. We've just sort of proof of concept that the setup works at this point. We haven't got the really interesting learning experiments done yet. But we're thinking along those lines, and we're not the only ones that are thinking along those lines. And then ultimately, it would be great to start looking at a stroke model and asking, what's the ability to recreate either connections or even modules. That's jumper cable, yeah.
I kind of wonder along those lines if there might be something from your work that would even clinical application, now not necessarily devices stuck in the brain to do this, but maybe in the rehab protocols that are done now. Could this indicate maybe there's a better rehabilitation exercise set that would maybe enhance?
Right, and when I think, so we mentioned earlier before the talk that I thought a little bit about stroke rehab earlier, and one way to think about this problem is, why is it that some people get better and some people don't? And my very limited knowledge on this is that although people have done lots of studies looking at predictors, even the best predictors are only so good. There's a lot of variability that's hard to explain. And what I think is missing and is very hard to quantify is what I would think of as kind of the state of learning. Okay, what I mean by that is you can imagine that you're in this big, the brain is a state. And there are good states, and you could think of those being low energy, down a hill. And there are bad states that are up, right? And what we think of, when we think of learning, a lot of what we think of the dynamics of learning is kind of going downhill to get to the good spot. Now, you can be in a place where there's a path to get downhill. So with simple learning algorithm, what most learning algorithms do in the machine learning world is they go downhill. It's a boring thing, but that's what they do, gradient descent. That's how they get better. And so most of what we think about the way the brain gets better is it incrementally gets better looking for downhill. But you can imagine getting stuck in a place where you're in a trough, you're in a valley that's just a bad valley to be in. And there's no easy route to get downhill because of an earthquake, whatever, right? And in that point, you've got a problem. You can't get over that hill with simple learning algorithms. You could just be stuck. So it might be that the reason why sometimes you don't get improvement isn't because the neural circuitry is broken, but because it's in a state of plasticity that's just too hard to get across. And in those scenarios, you can imagine that what you need is something to jumpstart you, to get you, to leap you over that mountain, and then you can learn on your own. And so some kind of almost disruptive version of going into the brain, stimulating, creating the right connections can get you over that hump. And then ultimately you might be able to remove that device altogether and be fine without it. So that's one version of what Dustin's saying. But then even before we get to that sort of more far out version, I think what I am trying to say is that thinking about the psychophysics and thinking about the role of correlated signals in the brain can be very important. Now, we're not the only people to think about that. People are thinking about, for example, passive movement as a therapy. And there what you're getting is combined vision and proprioception with a task. People have tried these sort of Hebbian approaches of stimulating during movement. And there's, again, the kinds of things that we're talking about. But trying to optimize those in the context of these learning algorithms, I think is something that would be interesting to think about.
So I really like your coherence task, and I'd actually like to try it myself. See
I have a little time. You're welcome.
But my question is that your ICMS is basically based upon a global error vector, so a known target.
Yeah.
How would you use that same paradigm in a situation, in a clinical BMI where you don't know the target?
Yeah, that's right.
How would you map your students?
So as I was saying earlier, the paradigm that we've got here that I told you about, the published paradigm, it's not specifically a model of BMI. So but the way to think about it is, look, what did we do? What we did is we stimulated, and that stimulation correlated moment by moment with the visual cue. But the problem is the same in the context of real BMI. So what Joey's doing right now, Joey O'Doherty in my lab, is he's got a monkey who's controlling a cursor on a screen with decode, from decoding motor cortical activity, BMI mode, controlling a cursor on a screen. So he's seeing that. So now the parallel's, I think, fairly straightforward. He's seeing that pattern. We're going to stimulate, and hopefully what will happen is that combination of those cues will be enough to drive learning of that signal. So this was not that, but we think the leap to that is not conceptually difficult. Now, it's a different paradigm. It's BMI. It's not natural control. Will it work? We don't know. We will know soon because we're doing those experiments literally. Well, it's still pretty early in San Francisco. Not literally right now, but later today.
Very interesting. I have many questions, but I'll ask you too. I see that you're discussing combination of peripheral and central stimulation. So maybe that peripheral stimulation is something that will help the brain get over the hump and get to, some studies with transcranial direct current stimulation showing that you need that peripheral input to sort of put the brain in the right state.
Absolutely, that's absolutely right, yeah. I mean, a really simple example of that, it's a kind of maybe a trivial example, but it's still really compelling, is the Ramachandran, phantom limb pain. You start moving it with the mirror and suddenly things feel better. And Dustin's got a version of that too. He was telling me earlier that you can get the phantom to move from sort of living on the stump to back to a more normal position when you start providing somatosensory feedback to a prosthetic hand. So that's not quite what we're talking about, but it's a good example where it's not that the subject probably didn't wish that phantom pain to go, or didn't wish to have a more normal percept, but without that kind of, without that natural feedback, it wasn't going to happen. But then once it's there, it's easy.
Right, yeah. And my other question is about the dose. So dose of stimulation, dose of rehabilitation, dose of relearning, re-education. You've done many experiments and you're looking at different areas in the brain. Do you see the difference in the dose of training required for monkeys to learn to use the cues?
Right, so that's something that we really, and that's kind of that rate of learning issue. And that is something that we simply have not yet quantified. We're very interested in the topic, but you know, we've only done these experiments, just sort of the data set that's published. So that's still an open and very interesting question.
Because
It's clinically very relevant. If it takes 3 years to rehab, you know, who's going to adopt it, right?
Do I need to have 5 treatments, or do I need 10?
Right.
I mean, that may
Right.
make a big difference.
Absolutely, yeah.
All right, well, I thank everybody for their attention this morning, and I thank our speaker one more time.
Thank you.